Thank you. So thank you, Meredith, for the introduction and for the invitation to come and to speak with you all about some of the research that's been ongoing in my lab for a number of years now. So what I thought I would do is introduce a little bit of the background, a little bit about P53 and how we see P53, the kinds of mutations that occur in human cancers, and the mouse models that we've generated to ask questions about how P53 functions and what mutations do when they're present in human cancers. So if there's something that's not clear, please feel free to raise your hand during the talk and I'll clarify whatever needs to be clarified. Okay, so I'm going to talk about the P53 tumor suppressor pathway. And so my first slide is just a slide that introduces the pathway. And this is a very simplified slide because it is a complicated pathway. A lot of things impact P53, and then P53 can do various things to the cells. So first and foremost, P53 is a transcription factor, and it's present at very low levels in a normal cell. And it's not really functional in a normal cell. However, under a number of different circumstances, anything that damages a cell, if you go outside and you get some UV light on your skin, X-ray radiation, hypoxia, anything that the cell senses is just not quite right, what happens is the P53 levels shoot up and it becomes a very stable protein. And in that scenario then, it can activate different genes that have different functions in a cell. So some of the first, well actually the first P53 target that was identified is called P21. And P21 is a cell cycle inhibitor and it induces a senescent program. Okay? Other genes that P53 activates, PER, BAX, these are all genes that are important effects of apoptosis, cell death. Okay? In addition, P53 can alter the metabolism of a cell, and so it needs different nutrients to grow. So these these three effects of P53 activity, the arrest, senescence, apoptosis, and the changes in metabolism are all critical for suppressing the cell, for preventing that damaged cell from replicating. The positive effectors are things that phosphorylate P53. Two important proteins on this slide that you'll hear about in a few minutes are these two proteins called the MDM proteins, MDM2 and MDM4, and these are absolutely critical inhibitors of P53. Okay. MDM2 has a special relationship with P53 because MDM2 is also a target of P53. Okay, so here the cell is damaged, P53 levels have to come up. If P53 kills a cell, well, it's out of the picture. But if it arrests a cell and then realizes that damage has been corrected or the cell now appears normal, then what P53 does is it turns on the MDM2 gene, which makes the MDM2 protein, which then dampens P53 levels and returns everything back to basal levels. Okay? And in that scenario, then P53 leads to survival of the cell, a survival of a normal cell. So because P53 has this activity, because it responds to abnormal cell behavior, responds to DNA damage, and it can induce this arrest, senescence, and apoptotic phenotypes, it's a critical tumor suppressor. And it is mutated in almost all cancers. So what I'd like to show you next is just a line drawing of P53. So it's about 393 amino acids long. It's a transcription factor, so here at the end terminus of the protein is the transcriptional activation domain. In the middle is the DNA binding domain, and at the end is a tetramerization domain. So P53 actually forms a tetramer to bind DNA and to activate transcription. And all of these little dots are mutations in p53 found in human cancers. The black dots are truncating mutations that truncate the protein and lead to loss of function. The green dots, which occur mostly here in the DNA binding domain and a few in the tetramerization domain, disrupt tetramerization and DNA binding. And these are missense mutations. The two that I highlight on this slide are the R175 His mutation, which is a very common hotspot. It's found in about 6% of human cancers, and another mutation in amino acid 248. Both of these are arginines, and both of these are critical for the protein to bind DNA. So you can see from the onset that these mutations are going to disrupt p53 function, but the protein is still there. And one of the topics that I'll discuss with you today is what that mutant protein is doing in the cell, because it's contributing to a very aggressive nature of tumor cell development. So p53 is so critical. I'm going to make this blanket statement that may not be true, but I think is true. And I think that the p53 pathway has to be dampened or inactivated or eliminated somehow in every cancer cell that grows. It is such a potent transcription factor. It is such a potent inhibitor of cell growth that I think that pathway has to be wiped out in every cell. I may be wrong, but there are probably very few tumors that can grow and have a functional wild-type p53. So what this slide shows is the relationship between p53 and the two proteins that I told you were p53 inhibitors, MDM2 and MDM4. No, I only have MDM2 on this slide. Okay, so let's go through this slide. Here on the left is an example of a tumor that's called a high-grade serous ovarian carcinoma. 98% of these tumors have mutations in p53. And in fact, if you talk to the clinicians, they think those 2% that don't have p53 mutations have actually been misdiagnosed. So in this cancer type, p53 mutations are rampant. Here, in liposarcomas, they have no p53 mutations. In fact, what they do is they amplify this protein called MDM2, which is a p53 inhibitor. Okay, so this cell has a completely different mechanism of inactivating p53. It makes very high levels of the inhibitor. And these cells have wild-type p53 by sequence. Okay, and if you compare these two, let's go to the glioblastomas next. So glioblastomas are different, okay, and they're different because they're a complete mix. So here in blue are the about 1/3 of glioblastomas that have p53 mutations. In red are about 1/4 of tumors that have either high levels of MDM2 or MDM4, the other inhibitor, and then about a little less than half don't have mutations in either MDM2 or p53. And I think that these cells have a different mechanism of dampening the p53 pathway, and it's an area of keen interest right now. We're trying to identify what's wrong with these cells. How have they inactivated the p53 pathway? But what I show here in the bottom part of this slide is a comparison of the different p53 and MDM2 mutations in this glioblastoma. And you can see that if the tumor has p53 mutations, it doesn't have MDM2 amplification. And if it has MDM2 amplification, it doesn't have a p53 mutation. These are mutually exclusive events. Okay? So if you look at other tumors and you look at breast tumors, for example, also have high amplification of these inhibitors, MDM2 and 4, and p53 mutations, you will see the same relationship. So what that suggests is that there's at least 2 different ways which a cell can get rid of p53 function. This is now a pedigree from a family that my colleague Louise Strong at MD Anderson has collected for now almost 40 years. And these are individuals that inherit a p53 mutation in their whole body. And if they inherit a p53 mutation, they are highly prone to getting all kinds of cancers. So let's just go through this slide. Any individual that is in red is a person that has a p53 mutation, and they are also have a tumor or tumors of sorts. The individuals in pink are unaffected individuals. They have a p53 mutation, but at the time of this pedigree in 2003, they do not yet have cancer. So if you look at the pedigree, you can see some amazing details. Here's an individual, a woman in fact, who had 5 different cancers throughout her life. So these individuals come to the clinic and they know they have a p53 mutation, tumor is identified, it's removed, and then they come back a year later or 2 years later and there's another tumor that comes up. These families are incredibly susceptible to cancer. The kinds of tumors they develop is just all over the board. So here you have breast, ovarian, gastric cancer, prostate, in this male, soft tissue sarcomas, lung cancers, melanomas, more breast, soft tissue sarcomas. So almost any cancer type that you can think of, these individuals can develop. And again, it emphasizes the universality of mutating p53 in order for a tumor to develop. So a few years ago, with the help of Louise, I updated this slide. So this is now a few years later, this is 2012, and what I want you to focus on is these individuals in pink. So in 2003, I think was the first slide, they didn't have cancers, but now 10 years later, you can see this person has a breast, another breast tumor at age 34, colon cancer at 50, breast 52, colon cancer 62, another breast, and another breast. So in that 10-year window, these individuals that had a mutation but no tumors have developed cancer. There's a wide range of age at which these individuals get cancer as well. So here, for example, you have a 2-year-old with a brain tumor, and up here you have a 61-year-old. So they're very complex families. We know that p53 is the driving mutation that leads to cancers, but we never know when and where those cancers are going to develop. So a number of years ago now, we decided that we wanted to generate a mouse that mimicked the Li-Fraumeni syndrome. So this would be a mouse that would inherit a p53 mutation throughout its body. And so the mutation that we decided to make is at arginine 172, which is the arginine I mentioned just a few slides ago, 175 in humans, which is highly prevalent and a very aggressive. From the studies that had been done in tissue culture, this was a very aggressive mutation. But we decided we wanted to study this mutation in vivo in the context of the whole mouse. So we knocked in a single point mutation that changes this arginine to a histidine. Sometimes we call this the H mouse, so if I use some jargon, this is the H mouse. This mouse expresses that mutant p53 protein. It has multiple cancers that it gets. It'll get sarcomas, it'll get carcinomas, it'll get some lymphomas, and in fact, depending on the background strain of mouse that we use, we can get some mice that develop breast cancers and others that don't develop breast cancer. So there's a whole range of tumor types that these mice can get. The most fascinating aspect of this mouse is it had a highly metastatic phenotype. So here, for example, is a tumor, and it is stained with the p53 antibody, and you have a lot of p53 mutant protein in this tumor. And here you have this huge metastatic lesion on the liver, and this is a lesion in the brain that again has been stained with the p53 antibody. Okay, highly metastatic. In comparison to a p53 null allele, which had been made earlier by Tyler Jacks and Larry Dunhauer, loss of p53 also contributes to a tumor phenotype, but not a metastatic phenotype. So the big difference here was that this mouse had a metastatic phenotype, which is actually what kills the human patient. It's not the primary cancer, it's the metastases that spreads everywhere. Okay, so anytime we've made a mouse, there's always some unexpected findings that you can either choose to study or not. And so we had 2 unexpected findings with this mouse. Here's the first one, okay? So here I'm comparing a mouse that has this p53 missense mutation to a mouse that has 1 allele missing, so it's a heterozygous mouse, and I'm plotting the survival of these mice, okay? So at the start, you have a cohort of mice and all the mice are alive, and then you put them on this graph as they start to die. So what can you see from this graph? There's absolutely no difference in the survival, whether the mice inherit this null allele or this mutant allele. Okay? But I just told you that these mice had a highly metastatic phenotype, so they should have died sooner, but they didn't. So in a few minutes, I'll tell you what we've learned about this model and how we've interpreted these data. Initially, we thought that because the veterinarians who watch the animals for us, they tell us when we have to sacrifice the mouse. And so one of the ideas here was that the mouse was sacrificed because the primary tumor got very large, and so that was the cause of death, and that's why this curve wasn't different. But you'll see that wasn't the correct answer. Now there's another finding that we didn't understand initially, and that's on this slide, okay? So what we've done here is we've taken tumors from these mice, and we've stained with the p53 antibody to detect p53 protein. So why do we do this experiment? The reason we did this experiment is because in human cancers, people stain for p53 protein all the time, and every tumor that has a p53 missense mutation has a stable protein. At least that's what was thought. But in this experiment, what we show here on the left are 2 tumors that are negative. Okay? So even though they express that mutant p53 protein, they're not expressing it at high levels as these 2 tumors are. Okay? But we've always thought that p53, mutant p53, was a very stable protein. And what this experiment suggests that it isn't a stable protein. Okay, so the next experiment that we did then to try to figure out what was happening with mutant p53 is to just look in vivo at a normal mouse. So we're not looking at tumors right now, we're just looking at tissues from a normal mouse. And what you don't see on this slide is any staining for mutant p53. So in fact, it's a very unstable protein, just like wild-type p53 in a normal cell, which was a surprise. We always thought it was going to be a very stable protein. So what is regulating that stability then? I'm going to introduce the MDM2 model that we also generated a number of years ago, okay? And I've already told you MDM2 is a potent inhibitor of p53, okay? So we made or tried to make an MDM2 null mouse. That means a mouse that has absolutely no MDM2 in it, and we couldn't. This mouse is dead at a very early embryonic stage. This is an embryo we call a blastocyst. It has about 128 cells at this point. It's just prior to implantation in the uterus. And we've stained this with a tunnel assay to detect apoptotic cell death. And you can see in this embryo that every one of those cells is black, every one of those cells is already dead. Okay? This was a complete surprise. Again, we'd never thought that that losing MDM2 would have such a dramatic phenotype on development. So the question we asked is, what happens to that embryo if you also take away p53? Okay? I've already told you MDM2 is a p53 inhibitor. So the hypothesis that we're testing here is that this MDM2 had such high levels of p53 that it was killing all those cells. Okay? So we do a genetic experiment, we do these crosses, and lo and behold, we can make a mouse that has no MDM2 and no p53. So what MDM2 is doing in vivo in a normal vertebrate is to dampen p53 levels. And when it's gone, p53 is very high. Now the reason I'm introducing this mouse to you is because we're now going to test the hypothesis that perhaps MDM2 is also contributing to the stability of that mutant p53 protein. Okay? What I haven't mentioned yet, and I will now, is that MDM2 is an E3 ubiquitin ligase, and it targets wild-type p53 for degradation. Okay? So now we think that it might also be targeting the mutant p53 for degradation. So here's the experiment. We know MDM2 inhibits and degrades p53, and so we're going to ask now if MDM2 degrades that mutant p53 protein. So this is a big experiment, and we had some reasons to believe that the experiment might work. And one of those reasons is that when MDM2 is discovered as a p53 interacting protein, the cell actually had this mutation in it. So we already had some evidence that this MDM2 could bind the mutant p53 protein. So this is just to show you where MDM2 and MDM4 bind. This is the p53 line drawing, similar to what I showed you earlier. This is the DNA binding domain. This is where all those missense mutations occur. This is where MDM2 binds. It binds the transcriptional activation domain and prevents p53 from functioning as a transcription factor. In addition, it targets it for degradation. Okay? So we had lots of reasons to believe that MDM2 might also be regulating the stability of that mutant p53 protein. So what do? I'm a geneticist. We do a cross. And so we asked what happens when you have the MDM2 null mouse and you have a homozygous mutation in p53. So the first thing that happens is you rescue the phenotype. Okay? So a mouse that has no MDM2 and has a homozygous p53 missense mutation is alive and well. Okay? So this tells you an important thing already, and that is that this missense mutation is functionally null. Okay? So now what happens to the stability of that mutant protein? So here again we stain different tissues with the p53 antibody. In the top panel we have the just the mutant mice, and you see little, if any, stability. This is some background staining in the intestine, but you can easily see here in these tissues that have no MDM2 and they have that p53 missense mutation, you see a lot of stable mutant p53 protein. Okay? As an aside, to me it's very interesting and we still don't understand it. So if you just look at this image, you've got maybe half the cells there that don't have stable mutant p53. So why is that? Is it because those cells will not upregulate p53? Is there another ubiquitin ligase that's regulating that mutant in other cells? We don't know. But what we do know is that MDM2 is regulating the stability of that mutant p53 protein. So we can make these mice. We know we've stabilized the mutant p53 protein, so the question is, what happens to the phenotype of these mice? And this is the survival curve that shows that having that stable mutant p53 protein is worse than not having p53. So here is a line drawing in black, the survival curve. So you start out with 100% live mice, and within a year, everyone's dead. These mice are homozygous for the p53 missense mutation. If I plotted the null allele here, it would be identical. If I plot the MDM2 p53 double null, it would be identical. And here's what happens with the mice that have no MDM2 and have that missense mutation, and these mice are dying sooner, and they're dying of a more aggressive cancer, and that is highly metastatic. So the homozygous mutant mice, we're looking just at sarcomas, so we're comparing apples to apples. We do not see metastases in the homozygous mutant mice. I told you that this was a metastatic allele, but that's in the heterozygous condition, okay? And again, we thought that it's all about the timing of tumor. if the tumor happened too quickly, if it developed too quickly, it wouldn't have time to metastasize. Okay? But what we've done here is we've taken out MDM2, we've stabilized that mutant protein, and now we get extensive metastases. So it's not that the window is too short for metastases, the window naturally is too short to stabilize that mutant p53 protein, which then leads to a metastatic phenotype. I think, yes, okay, and in a few minutes we'll discuss the model again. Because it's important to understand in human tumors what leads to stabilization of that p53 mutant protein, and then how that protein contributes to a metastatic phenotype. So what I've shown you is that MDM2 regulates wild-type p53. It inhibits wild-type p53. It also inhibits the R172H mutation. MDM4 is a related protein that's also an inhibitor, and we also have data that says that this protein can also regulate both of those, can also inhibit both of those, okay? But the next question we wanted to ask is a little bit more broader. Okay, MDM2 regulates these proteins, but MDM2 is something that we can play with in the lab. Well, what about things that naturally stabilize wild-type p53? Ionizing radiation, oncogene activation, stress, any kind of abnormal cellular behavior. So in the next two slides, I'll show you how DNA damage and reactive oxygen species stabilize that mutant p53 protein. So here's the first gel. So what we've done here is we've taken mice that either have no p53, have this missense mutation, 2 alleles, or have the missense mutation and don't have MDM2, okay? So if you look at p53, the mouse that has no p53 has no p53 on our Western, nice control. Here is the homozygous mutant mice. Before DNA damage, before irradiating this mouse, you don't see p53. You irradiate the mouse and you get very high levels of p53. And if you take out MDM2, you have very high levels of p53 to start with. Okay, so this experiment tells you that radiation something is sensing that damage and is telling the cell, I need p53. So p53 levels go up, but the cell doesn't know that that p53 is mutant. Okay? Now the other thing I want to point out from this experiment is that each one of those lanes is a whole mouse that we put in an irradiator and we irradiated it. Okay, that's a lot of work to get all these genotypes and to irradiate the whole mouse. So the question that you should be thinking about is, why didn't you just take cells from these mice, put them in culture, and look for p53 stability, mutant p53 stability? And the reason we tried, but the reason we can't do that experiment is as soon as you take cells from these mice and you put them in culture, it's a stressful condition. The oxygen tension is different, the media has all kinds of stuff in it. The cell is stressed out and it stabilizes p53, mutant p53 and wild-type p53 almost immediately. So we have to do these experiments in vivo in the whole mouse. Now the other experiment I want to show you is this reactive oxygen species. We've known for a long time that ROS stabilizes wild-type p53, and so the question we asked here is, does it also stabilize mutant p53? So here we have 2 lanes each. This is the p53 levels in these homozygous mutant mice. You irradiate, you stabilize that mutant protein. And then we added N-acetylcysteine. And this is a drug compound that squelches reactive oxygen species. And so anytime we add NAC to the mouse, you see that you dampen the p53 levels, both in the spleen and in the thymus. And the reason we use these 2 tissues is because they both express very high levels of p53 in response to radiation, and we can detect it very easily. Okay, so I've shown you 2 different damage signals, radiation and reactive oxygen species, and they're both stabilizing mutant p53 as effectively as the wild-type p53. And so we've tried a few other things, and everything that stabilizes wild-type p53 stabilizes mutant p53. And so we will, we're not going to do any more experiments. I think that the data are pretty consistent that you damage the cell, and the cell senses that damage, wants to generate some wild-type p53, but if it's mutant, it's the mutant that's going to become stabilized. Okay, so let me summarize what I've told you so far. Mutant p53 is an inherently unstable protein. This was completely opposite of what we thought before we'd done these experiments. Tumor-specific alterations, so something is happening in that tumor that is stabilizing the mutant p53 protein. Once stabilized, then, it can show these aggressive phenotypes, which in vivo is an invasive metastatic phenotype. The mutant p53 stability is also regulated like wild-type p53. So the same kinds of signals that will activate wild-type p53 will activate mutant p53. So here's a model. And the aggressive nature of the tumor is going to depend on when that mutant p53 becomes stable and how it becomes stable. And this is a real, I think, important question in the literature. So what I've shown here in the top panel is a cell that's normal that stabilizes p53 very early and leads to a very aggressive cancer. And this is reminiscent of what we see in ovarian carcinomas. Okay? 98% have p53 mutations, very easily detected by immunohistochemistry. So something in those initial cells, we don't know if it's genomic instability, a stress signal, something is stabilizing that mutant p53 protein and leading to a very, very aggressive cancer. In the second panel, what I've done is I've shown you an example of a colorectal cancer. So colorectal cancer, the first mutation is usually a RAS mutation, and the last mutation is a p53 mutation, okay? But it's when that tumor has a p53 mutation, it's a very stable protein, and it's a very invasive from that point forward. And then here, I'm just proposing that maybe there are tumors out there that might have a p53 missense mutation, but didn't stabilize it for whatever reasons. Maybe it's a very slow-growing tumor. We have lots of tumors that are very indolent and grow very, very slowly for a long time. And those tumors might have not stabilized that mutant p53 protein. And so it's just kind of managing to survive. So there's lots of things that have been published about the mechanisms. How does this p53 mutant protein contribute to this invasive behavior? And I'm just going to list some of these here because I won't have time to discuss them. But basically, when you have a stable mutant protein in the cell, you've now created the potential for protein-protein interactions. Okay? And so we know that this mutant protein can bind p63 and p73, which are also tumor suppressors. We know that it can bind integrin and affect integrin recycling, which also affects the membrane's invasion. But we also know that this mutant p53 interacts with a series of transcription factors and modifies what those transcription factors are activating. So here's an example. SREBP is a transcription factor in the mevalonate pathway. And Carol Privis's lab published some beautiful data that shows that mutant p53 can bind SREBP and activate this program. Okay? Now there's two important things to note. Number one, p53 can't bind DNA directly. It has mutations in the DNA binding domain. And so it usurps other transcription factors to activate transcription. In addition, this mutant protein, because it has mutations in the DNA binding domain, has an intact transcriptional activation domain. And so basically that transcriptional activation domain drives these different pathways. And that's what contributes to a very invasive and aggressive cancer. Okay. So the mouse that I've been describing to you that we've studied for a long time is a mouse that has a germline mutation. Every cell in that mouse has a do you have a question? Oh, okay, you're okay. Every cell in that mouse has a p53 mutation. okay? And this is very reminiscent of the Li-Fraumeni syndrome patients who inherit a p53 mutations throughout their bodies. But what we really wanted to study is a somatic mutation, and that is all the cells are normal, but one cell expresses that mutant p53 protein. Okay? And these are most of the cancers that clinicians see in the clinic. One cell has acquired a p53 mutation, and it's the cell that becomes the tumor, and everything else is normal. So there's different mice that we and others have been studying. So this is the little pink mouse here is the germline mouse with the mutation in every cell, highly metastatic tumors. This is a mouse that my colleague Tyler Jacks at MIT made, and it was a cool mouse about 10 years ago. And it can generate a p53 mutation in a specific organ. So he has studied lung cancer, and he studied pancreatic cancers, because those cancers have p53 mutations most of the time. But the way he manipulated the genome, the whole mouse is heterozygous for p53. So here in gray then, the mouse has one wild type and one null allele. And then the mutant has the pancreas or the lung has a p53 missense mutation. So it's not really a somatic model. What we wanted to make was a mouse that had this p53 missense mutation driving lung, breast, pancreatic cancers in a context where the rest of the cells were completely normal. Okay? And this will allow us to examine how the immune system contributes to the development of the tumor or the inhibition of the tumor, and the stroma and how it affects the tumor environment. So we really wanted to make something that was more reminiscent of what the clinicians see in the clinic. Okay, so here's how we generated it. Okay, so here's the concept. We have an allele that contains that p53 missense mutation, the same one that I've been describing. In addition, though, prior to the mutation, it contains a cDNA insertion that allows expression of a wild-type protein. Okay, so this allele expresses a wild-type p53. There's a polydylation site here, so translation stops here. Now, this allele, the cDNA portion that's wild-type, we flanked with what's called a LOXP site, okay? And this is just a means by which we can use this bacterial protein called Cre recombinase to delete anything in between those two little triangles, okay? So when we have Cre, we delete this blue sequence and we make a protein that is a mutant protein, okay? So all we have to do now is combine this p53 this p53 allele, which we call WT for WM, wild type to mutant, okay? So all we have to do now is have this allele present in our mouse and then express this Cre recombinase, and we can make just a few cells express the mutant protein. Okay? It took a long time to make this mouse. And so the first thing I'll tell you is that we actually made 2 mice. Okay? Actually, Yuan Zhang in the lab made 2 mice. One was the same mutation that we've already been discussing. And when we remove that wild-type cDNA, this allele is identical to the one we made in the germline, okay? And we thought this was important because we didn't want other things to contribute to maybe differences in the levels of the protein. And then we also made this other allele at 245, which is at 248 mutation in humans, which is also another hotspot mutation. And I have to, as an aside, again tell you that 3 different labs tried to make this mutation about 15 years ago, and we could not make this mutation. We still don't understand why. But in this case, we were able to make the mutation because we had this wild-type insertion, and so the allele makes wild-type protein, not the mutant protein from the get-go. Okay? So What we've done then with these alleles is, the first thing we've done is to check and make sure that this WM allele is as wild type as it gets, okay? So let's back up for just a second. So what we're doing is we're characterizing this allele, and we're characterizing this allele to see how wild type they are. Okay, so in this experiment what we're doing is we're irradiating cells. We know if we irradiate cells, p53 levels become stable, and p53 will activate transcription. So what you can see here is that in these R172WM cells with the 172 mutation, p53 is stabilized. The 245, it's also stabilized, although not as well. But here we looked at two p53 targets, p21 and PUMA, and you can see comparison to wild type or to our allele, there's no difference. Okay, so the allele looks like a wild type p53 as far as transcription. What about some of the other functions? Apoptosis, cell cycle arrest are two other effects of wild type p53 activity. So here you see what happens to wild type p53. And this is in the thymus, and we irradiate the thymus and we get rampant apoptosis. It's p53 dependent. So this is the levels of apoptosis in the wild type with the H allele and with the W allele. There's no difference. Okay? So when these mice get irradiated, all of these three genotypes have the same apoptotic response and the same cell cycle arrest program. Okay? Now the million-dollar question is really what happens to tumors. So if this allele is truly wild type, it shouldn't get tumors, which these mice, if they're normal, it takes about 3 years to get a tumor. So that's a very long time. So what we've done in this case then is we've taken all three of those alleles, the two alleles, the wild type allele and the WM172, and we did it for the 245, but I'm not going to show you that data. And we added a null allele. So now it's a heterozygote. Okay? So now there's a tumor phenotype. So these mice in blue are the p53 heterozygotes, and then in red are the WM allele with a null allele, and there's no difference between the survival of those two mice. So it suggests that this allele is as wild type as it gets. These are the different kinds of tumors that develop in the two different mice, and there's no difference in the kinds of tumors these animals develop. And then this is the real experiment, which was a we've been working on this alleles for about 5 years now, and so we did have this cohort in the background. So this is now just comparing that H allele to the H allele and the 245W allele and a wild type allele, and those mice are getting cancers at the same time. 800-day experiment. So you just put these mice on the shelf and you watch them every day, and 3 years later you have data. Okay. What this suggests, though, is that there's actually no difference between our alleles that we generated express as much wild type p53 as they should express to inhibit tumor growth. Okay. So there's a couple of experiments we've started, and I'm going to tell you about one of them. And I think I have just a few more minutes. Okay. So I have a terrific postdoc, Yun Zhang, who generated these models. She was so afraid that she would make this mice put the p53 mutation in a few cells and never get a tumor, okay? Because we're comparing it to a mouse that has p53 mutation in the whole body. So the experiment that she did very tentatively was we had the mice with the R172H and the R245W alleles, WM alleles. In addition, in the background of these mice, we used this other allele, which is called TD tomato. Okay, and what she did is she wanted to look at breast cancer, and so she learned how to do an intraductal injection into the mammary gland of a virus, adenovirus expressing Cre recombinase. Okay, so what happens then, and I'll show you a pretty picture in a minute, is that you generate the p53 mutation in a cell, because Cree will recombine out the wild-type sequences. In addition, this TD tomato also has a Cree lock-stop-LOX allele in it, so if Cree is functional, the cells will turn red. Okay, here's a picture. Picture's worth 1,000 words. So here, Yuen used 2 different doses of virus. Again, because she wasn't sure. This is the experiment we really wanted to do. Just a couple of cells would have the p53 mutation, okay? But again, because she didn't think that those mice were going to potentially get tumors, she also used a higher titer of the virus, and in this scenario, 50 to 70% of the ductal cells are positive for the mutation, and they're fluorescing red, okay? An important, critical in these experiments is that the stroma and the surrounding tissues are normal. They don't have the p53 mutation. So this is actually reminiscent of what happens in the clinic. Okay? The last thing I'll add here is that normally the strain of mice that we use is called a C57 black 6 strain. It's just the genetic background that we've been using forever. A C57 mouse is incredibly resistant to breast cancer. So we never thought we would see breast cancer in that strain. So what we've done here is actually generated an F1 hybrid with a BALB-C mouse, which has a tendency to get breast cancer. So we're trying to make the mouse get breast cancer. Unfortunately, the experiment worked. So there's a whole lot of data on this slide showing the results of the mouse with the R172H mutation and the 245W mutation. So let me walk you through it. Okay, so here, the H allele was the first one that we generated. So Yuen added the adeno-Cree virus into the duct. She used the 2 different doses that I showed you on the previous slide. And within 1.5 to 2 years, she looked for tumors, and the mouse that had the low titer never got tumors, but the mouse that had the high titer did get a tumor. We got 1 tumor. Okay, again, because we didn't know if we were going to get cancer in this model, we took an additional cohort, and what we did was we gave them a sublethal dose of ionizing radiation. And we've known from previous experiments in the lab, if you give the mouse this low-dose ionizing radiation, it causes enough breaks in the DNA and mutations that the mice will get tumors earlier. And sure enough, the same 2 doses of Cree virus, now with the sublethal ionizing radiation, gave us a lot more tumors. So we got 1 tumor with the low dose, and we got 5 tumors with the high dose. Okay. Now the interesting experiment happened. So here we used the other allele, the 245 allele. Again, low and high titer, and within a year, these mice got lots of tumors. So the low titer, we had 4 tumors, and the high titer, we had 8 tumors. And the number in parentheses are the numbers of tumors that metastasized. Okay, so 2 or 3 take-home lessons from here. The R245 allele is much more potent at generating tumors than the 172 allele. And the 245 mutation readily gave rise to metastatic lesions. Okay, so here is the data presented a little bit differently. So here is again the tumor incidence based on the previous table. So the mice that just had the H allele never got tumors, except for that 1. With ionizing radiation, you saw tumors, both low dose and high dose. With the, here's another mouse that we made. So in this scenario, we took the H allele over a null. So we basically made the mutation and took out the wild type allele at the same time, only in the breast epithelial cells. And those mice got a lot of cancer. And then here's the 245W heterozygous mutation, and you can see that regardless of the dose of virus, these mice were getting a lot of tumors, and they were highly, highly metastatic. So the irradiated mice got metastases. I think we saw 1 out of 5, so it's a small percentage. The mice that had a mutation over no p53 also metastasized, but the 2, 4, 5 metastasized the most. Okay, and then here's another interesting tidbit of information. In humans, what happens is if you have this missense mutation, half of the time as a tumor develops, you lose wild-type p53. So the tumor only expresses the mutant p53. But in other cases, you keep that wild-type p53 allele, okay? So when you lose the wild-type allele, it's called loss of heterozygosity. So if you look at these H mice that were irradiated, 100% lost the wild-type allele. Okay, so we think that the reason these mice were so slow at developing cancer was that you had to get rid of the wild-type p53 for the tumor to develop. And that's in contrast with the 245 mutation. About half retained the wild-type p53 allele. Okay? So the way the tumors are evolving for these two different p53 missense mutations are really very different. And then this is just a comparison to human data, because we haven't published these last bit of data. And any time that you try to publish something in a field and it's in a mouse model, everybody wants to know human relevance. Okay, you made a mouse is not a human. You know, how relevant is it to humans? So what we did here is we just looked at data. There's lots of sequencing data available in the TCGA database. And you can look for, what we did in that database is we just looked at breast cancers, and then we saw some differences, but they weren't statistically significant. So here we're comparing breast, prostate, and lung, so all epithelial cancers, and we're looking at which ones have the H mutation and which ones have the 245 mutation, okay? And when you compare the survival of those groups, the individuals that have the 248 mutation equivalent to the 245 mutation that we just talked about have a poorer survival. So it suggests, again, the human data, which is association, is suggesting that the 248 allele is a more detrimental allele. Okay, the mice develop different kinds of cancers. Different kinds of breast cancers. So this is a moderate differentiated adenocarcinoma, highly anaplastic. This is a sarcoma-like, and then here I'm showing you the metastases, and we get mets to the lung and to the liver. Okay, the lower panel, what we've done is we've used a series of markers to try to identify what kinds of cancers these individuals get. And so the 3 genes that are used to clinically identify cancers are estrogen, progesterone, and HER2. These are hormones. The worst kind of breast cancer that women get are triple-negative breast cancer, and they have no expression of any of these 3 genes, 3 hormones. And you can see here in black that the R172H mice that were irradiated are mostly triple-negative breast, and with the 245 mutation we have about a third triple-negative, and then you have combinations of different other markers. So what the data suggests then is that the model is driving a breast cancer phenotype, and it's driving different kinds of breast cancer tumor development. And the last slide, which I don't have I'm actually out of time, I'll just show you briefly, is we've sequenced 2 of these tumors. And what we've learned from these is 2 things. Number 1 is we've learned that very early on in the tumor, in the initiation of the tumor, you already have metastases. And that we know because we can identify mutations in the mets and in the tumors, and we can figure out clonality and where they came from. And then the other thing we've learned is that, at least for the 2 tumors that we've been able to sequence thus far, is that they actually evolved in completely different programs. And both of these tumors have the same driving p53 mutation. So that's all I have to present today. Just sort of to overview, my interest has always been in trying to understand how p53 contributes to a tumor phenotype, and we've been using the mouse to model those mutations, and I think some of these new alleles are going to be very interesting to try to understand how the tumor interacts with the environment to grow and evolve. And last but not least, my last slide is the lab. Yuan Zhang and Xunbin Zhang are the two individuals who've actually worked very hard at generating that somatic model and the missense mutation. Jan Parker-Thornburg is the woman who runs our transgenic facility, and she makes all our mice for us now. Adel and James Yu are pathologists that help us with all these details. Patty Peyrong is here. Where's Patty? Not in the picture. And Kati are all technicians that help with all of the genotyping that we have to do when we do these crosses. So I'll stop there and I'll take questions. Thank you. Don't be shy. I'm sure it wasn't all that clear. Yes?
Oh, thank you. I want to know, how do you breed the mice themselves?
You put two together?
Well, I know you put two together, but I mean, like, do you have to do in vitro with each mouse, or do you actually just let them at it?
We let them at it. So, no, it's a good question. It's a very good question. So, one of the reasons the mouse is a terrific model is because 6 weeks after birth, it is sexually mature. And then gestation is 3 weeks. And then in 6 more weeks, those mice are sexually mature. So you can have a whole series of mice within 3 months to do the experiments that you want to do. But we can also, it's difficult. I mean, sometimes they don't breed. If there is noise in the facility, if there's construction going on next door, forget it, they're not going to breed. And so we have to cope with those kinds of issues. Another important point that I really couldn't point out because it would be too many more slides and too much work is that it's absolutely critical that you establish these cohorts at the same time, okay? So, and I can tell you I've run my lab for 30 years now, and we've had the p53 null mice in the lab for different experiments. And I can show you that experiments that we did early on versus experiments that we did today with those p53 null mice, and they are statistically different in terms of the survival and the tumor onset. Okay? So that suggests that there's variables, light, noise, music, I don't know, that are different at different facilities and that can alter the genotype. So one of the things that we do to generate a whole bunch of mice very quickly is we superovulate a female. So we take a very young female before puberty and we inject the hormones that cause all her eggs to ovulate at the same time. We isolate those, we fertilize those, naturally fertilize those eggs, isolate them, and then put them into 3 or 4 different mice, about 8 to 10 in each animal. And so in one big experiment, we can get 50 mice born. So we do all kinds of tricks to make sure that we get the mice that we need for the experiment within a relatively short period of time. Yes?
I was curious, so with p53 and its various downstream targets, are there like epigenetic factors that relate to which target is activated?
Great question. Probably yes. We've done a couple of experiments that suggest that the targets that p53 activate are different depending on the cell type. So we have some experiments in which we can activate p53 in the kidney, and we get p21 coming up 200-fold. It's incredible. But in the skin, we never see p21 come up. So for sure, there are factors that are either other transcription factors or epigenetic factors that will contribute to where p53 binds in a tissue cell type specific manner. Yes? I thought there was another question over here. Yes?
So one of the first slides you showed showed a very, very large number of liposarcomas having amplified MDM2.
Yes.
So one of the things that they found with that is that many of those MDM2 amplicons are sort of flung off in these extrachromosomal nuclear DNA
Bits.
fragments. And I know there was a big paper that was published earlier this year talking about those. Has anyone looked, do you know, to see if that MDM2 is being transcribed and is working
In those fragments?
in those extrachromosomal fragments, and to figure out if that's actually what's regulating that P53?
Okay, so I don't know if they've done it for that experiment, but I will tell you historically how we identified MDM2. So MDM stands for mirroring double minute. So MDM2 was identified in a cell line that had these extra chromosomal bits. And it's a cell line that had been in culture for a long time. Donna George actually cloned those double minutes, and she found 3 genes, MDM1, MDM2, and MDM3. And when she used those separately, MDM2 is the only one that gave the cell a growth advantage. So it's a cell line, it's not a human, but I would think so, but there's no data yet for the humans to show that, but that, yeah, that would make, yeah, exactly. Absolutely. Well, actually, in the clinic, there are now some MDM2 inhibitors. Really interesting. So what these, Nutlin is the first one, and so they're called Nutlins just because of the first one. And what the Nutlins do is they disrupt MDM2 from binding P53. And so the idea would be that for those tumors that have wild-type P53 and lots of MDM2, you can disrupt the interaction, reactivate P53, and kill the tumor cells. The problem with Nutlins right now is that there are so many adverse effects. And the reason is that you don't target it to the tumor. The whole body has increased levels of P53. And you can see from the mouse, the MDM2 mouse, null mouse that we discussed, and other mice that I didn't discuss today, but if you take out MDM2 in the whole mouse, in an adult mouse, that mouse is dead within 3 days. And it has a hematopoietic defect, it has kidney problems, it has almost every tissue we can find, liver problems, almost every tissue. So MDM2 is such a critical regulator of P53. To me, it's not surprising that we're having these toxic effects in the whole person. So I think what we have to do is figure out how to target these inhibitors specifically to the cancer cells or to a smaller number of cells, and not to the whole body.
Okay, let's give Dr. Lozano another round of applause.